Alright, I'll be presenting Lab Report 1 for BB2902, which is titled Induction of Beta-galactosidase. So this is a study of the regulatory genes involved in the metabolism of lactose in the bacteria E. coli. The genes involved are the lac operon, and the lac operon, or operons, are clusters of genes that are turned on or off at the same time, which is known as coordinate regulation. The lac operon has three genes that are coordinately regulated, which are LAC-Z, LAC-Y, and LAC-A, and they are controlled on the transcriptional level to be turned on when lactose is present in the cell. However, when a better source of carbon or no lactose is present, the operon is turned off. The genes are regulated by promoter, operator, and cap binding site regions of the operon located upstream of its coding region. The LAC genes are transcribed when RNA polymerase binds to the promoter region. The process can be repressed by two modes of regulation, which are negative regulation and positive regulation. In negative regulation, a protein called repressor binds to the operator region of the operon, which overlaps with the promoter region. This prevents RNAP from binding and transcribing the genes. When lactose becomes available, it binds to repressor and changes its conformation, inducing transcription. In positive regulation, glucose levels are low and cyclic AMP levels are high. The cyclic AMP forms a complex with a protein called CAP that binds to its binding site on the operon. This binding turns the gene on and induces transcription. The lac operon gene of importance for this lab is LAC-Z, which codes for the enzyme beta-galactosidase. Bacteria can have different genotypes, which is tested for in the second part of the lab, and the three genotypes are lac positive, lac minus, and lac C. So the objectives of this lab are to perform both a quantitative and qualitative assay on beta-galactosidase activity to see how quickly the lac genes are induced in the presence of lactose, and also to determine the genotypes of different strains of bacteria in respect to the lac operon. Hypotheses for this lab were that in the quantitative assay, enzyme activity for lac positive would increase until a peak of efficiency was reached and would then level off. Also in that assay, the lac negative strain was predicted to not increase much at all. In the qualitative assay, it was predicted that lac positive plates would grow blue only when lactose was present. Lac negative plates would grow white, and lac C plates would grow some blue when either glucose or lactose was present, because the operon is always turned on. So the lab follows the procedure that can be found in the document titled BB2901 Lab 1, which is Induction of Beta-galactosidase, written by Buchholz in 2009. The only exceptions to the procedure are that LB medium is used instead of sterile water as a blank in the optical density assays, and the LB blank must go through all the same steps as the test samples to make it an identical control that is only missing bacteria. So in the lab, bacteria that were both lac positive and lac minus were exposed to lactose and OMPG and allowed to incubate for 60 minutes. Spectrophotometer readings were taken at 20-minute intervals of both 420 nanometer and 500 nanometer wavelengths. The optical densities of the lac positive and lac minus strains of the bacteria are measured at 550 nanometer wavelength, can be seen in Table 1. So this shows that over time the optical density increases as the bacteria increase their activity. The optical densities were then plotted to be shown graphically, which is in Figure 1, and you can notice that lac positive gradually increases while lac minus stays roughly the same. The optical densities for both lac positive and lac negative strains can be seen in Table 2, and again it was graphed to show it more easily, and you can notice that there's a peak at about 20 minutes for lac positive. So then the data was normalized. Normalization allows to remove some interference caused by bacterial cells in the spectrophotometer. So this is first you see the formula for normalization, in which you take the optical density at 420, you multiply it by the optical density at 550, and then divide by 550, and this removes the bacterial cells from the equation. So normalized data can be seen in table 3, and again it was graphed to show it easier, and again you can see that lac positive peaks at 20 minutes and then levels off, while lac minus remains relatively constant. So in the second part of the lab, 3 unknown strains of bacteria containing different genotypes of the lac operon, the positive, minus, and the C, were plated on lactose and glucose plates. The resulting growth and color of growth can be seen in table 4, which shows that the sample plates for unknown A grew blue colonies on glucose plates, blue colonies on lactose plates. The unknown B grew half blue colonies and half white colonies on glucose plates and blue colonies on lactose plates, and the unknown C grew white colonies on both plates. In the final part of the lab, data was collected from the class and lab section, and it was statistically analyzed. The mean and standard deviation for both lac positive and lac minus readings at 60 minutes for both 420 and 550 nanometers were determined for both class and section. Can be seen in table 5. So this lab was a quantitative and qualitative study of the activity of the lac operon in E. coli. After performing the quantitative and qualitative assays of this lab, we were able to confirm both of our hypotheses. We were able to identify each of the lac genotypes on the plates, and we observed that enzyme activity of lac positive definitely reached a peak efficiency. We also observed lactose induction in the wild-type strain of the lac operon. This was determined because lactose was present in the medium for the cell cultures, and the resulting cultures cleaved OMPG, which indicates that the operon is activated because it changes color when it's cleaved. This was measured by the yellow color that results from the cleavage. The more induction that occurred is represented by a higher optical density reading, and optical density readings can be influenced by one of the samples having more bacteria. And since the experiment was run at 37 degrees Celsius, cell growth did occur. So when looking at the 550 nanometer optical density data, it is obvious that cells were growing because both samples of cell count goes up. And cell growth is the reason that data needs to be normalized, because normalized data accounts for them and removes any interference that occurred due to the number of cells that were present in the samples. We were able to find that the maximum efficiency was reached at about 20 minutes, where the peak in OMPG induction can be seen in both the original data at 420 nanometers as well as the normalized data. Also, when growing different strains of bacteria containing the lac operon, the following were the expected and observed results. The negative strain would not grow blue on either medium because there is no lac operon. The positive strain would grow blue only on the lactose plate because that glucose is present. The operon is, because when glucose is present, the operon is turned off. The wild-type strain has its operon at all times, so the culture is always has some blue in it. The statistical analysis of class data indicated that our 60-minute point was normal because it was in standard deviation of the class mean, and also being able to conduct a qualitative and quantitative assay on the activity of an enzyme's activity is important for determining the conditions in which that bacteria will grow. In particular, the lac operon allows the bacteria that has it to grow in the presence of lactose, but also does not function when a better source of energy is present. Different strains of the same operon also function differently and can affect where the bacteria can grow. References are Buckholt, 2009 BB2901, Lab 2 Induction of Beta-Galactosidase.